Welcome to Ask the Expert. We have with us Dr. Sangeeta Dhawan, coming to us from City of Hope in Duarte, California, Southern California. She's going to be speaking about beta cell identity from development to disease, and just a little bit about her. She's an associate professor in the Department of Translational Research and Cellular Therapeutics. She received her PhD from the Indian Institute of Science in Bangalore, India. She just studied developmental regulation. She did her postdoctoral training at Harvard Medical School and UCLA. And her training at UCLA focused on understanding the mechanisms of beta cell mass regulation, was supported by an advanced postdoctoral fellowship from JDRF. Dr. Dhawan has been a recipient of the Startup Award and the Smuckler Award for Diabetes Research from the Larry L. Hilbaum Foundation, a Transition Award from JDRF, and a Hearn, sorry, I'm adding more people, New Investigator Award. She served as a standing member of the ADA's Research Grant Review Committee, as well as an ad hoc reviewer on several NIH study sections. She currently hosts the virtual islet research seminar series, along with leading islet biologists from UCLA, Mayo, WashU, and University of Montreal. And she peer reviews for many high-impact journals such as Nature, NatureCom, Science, Science Advances, STM, and JCI. Oh, more, Journal Experimental Medicine, Diabetes, and Diabetologia. And she's an associate editor with Frontiers in Endocrinology. Research at the Dhawan Lab is currently supported by NIH, and her group is interested in understanding the biology of insulin-producing beta cells. Both type 1 and type 2 diabetes result from the loss of beta cells, making beta cell replenishment an important therapeutic goal. Approaches to this end include generating beta cells from stem iPS cells, as well as protecting or generating endogenous beta cells. Successful outcome of these approaches demands the generation of abundant numbers of functional beta cells that are survival competent in the diabetic milieu. So with this in mind, they're focusing on understanding the epigenetic and cellular signaling pathways that regulate the formation, function, regeneration, and survival of pancreatic beta cells during development and under circumstances that warrant the repair regeneration of the beta cells. We're very excited to hear your talk today, and welcome. Thank you so much for joining us.
Thank you, Monica, for inviting me. Thank you for all your efforts with Sugar Science, which brings the community interested in type 1 diabetes research. That's our interest as well. And in that realm, we focus on this really cool cell, that is the beta cell. We love islets. And of course, the interest comes from the fact that in diabetes, as everyone in this forum knows, there's a decline in functional beta cell mass, which comes from two components. One is the loss of function, whereby beta cells have blunted insulin secretion. They have loss of functional maturity. Their identity is impaired. They're no longer like their real selves. As well, there's a deficit in the total number of beta cells. So of course, there is cell death ongoing, but there's also a failure to regenerate beta cells, right? And for today's talk, I'm going to be focusing on this component of beta cell loss of maturity and impaired identity. Now, let's talk about beta cell identity. What is beta cell identity? I think the working definition of what a beta cell is that a cell that is producing and secreting insulin, right? And so the definition of a mature beta cell then is something that's responsive to the highs and lows in blood sugar levels. So if we look at what happens to a mature beta cell in the context of diabetes, there can be few different things that happen to beta cell identity. For example, there can be dedifferentiation, which is sort of like a time travel back in developmental time. So beta cells repurpose some of the programs that they once used during development. There can be transdifferentiation, beta cells switching or appropriating features of another related cellular fate, for example, alpha cells. Of course, there's the phenomena of senescence, which has recently gained a lot of interest in the field, whereby beta cells are just sheltering in place. They are experiencing a lot of stress. They are in the pro-survival mode, and they're not able to function well, and yet they're determined to stay put, right? So that's the senescence aspect. And collectively, all these changes in the beta cell identity cause the dysfunction of whatever beta cells are there in the diabetic milieu. And that also begs another very important aspect, that these things are not happening to all the beta cells. So there's a heterogeneity of beta cells, and there's altered heterogeneity. beta cells are not all alike. And this was recognized a while ago, but there's more and more interest in this now because we are, given the advances in technology, able to go very granular on this. So there is a lot of beta cell heterogeneity, which is an inherent aspect of beta cell identity. And this heterogeneity can come from a variety of factors. For example, the developmental state. There may be some types of beta cells that are more prevalent in early developmental states than in the late postnatal states. They can be there can be beta cell types based on their location. For example, Mark Housing discovered this very interesting cell type called the virgin beta cells, which are somewhat immature, but they're always located on the islet periphery. Different response to the environmental cues, which also ties in with the new idea of the functional phenotypes of beta cells. For example, the hub cells that are more connected to other beta cells, or the leader cells, the wave initiators, the first responders, which temporally respond differently to different type to the glucose stimulus, right? So a lot of work from Richard Benninger, Vera Kravitz, and Guy Rutter has talked about these types of different functional phenotypes of beta cells. There can be changes in morphology. For example, senescent beta cells look different. They're more stretched out. And then there's the question of the molecular profiles. Beta cells can have different molecular features that set them apart from other cells. There are cell types that are more stressed than the others, say in the context of senescence. There are some cell types, for example, the flat top positive versus negative cell types that have different proliferative potential. And then they can be gradients of transcription factors. For example, Dr. Hodgson's group has described this high-low populations that are more or less enriched in beta cell specific transcription factors that impacts their function, right? And then Andy Pospisilik's group now has come up with this very cool idea of epigenetic heterogeneity on how the epigenetic dosage of certain polycomb proteins can sort of distinguish pools of beta cells that are differently wired. And so there's a lot of heterogeneity, but there's a method to this madness. Nothing happens in biology without a reason. So it's really important that we understand what these different cell types are trying to do and what they're trying to tell us in terms of the biology of the islet behavior. And then there's the other question, which is a more esoteric question of, are these different beta cell types cell fates or cell states? Now, cell state would be something that is more transitory. For example, a cell that is replicating and then becomes quiescent, and a cell fate would be a more long-standing and more permanent sort of the state of the cell. For example, cells that may express something at a certain stage of development versus not, or certain beta cells that always express something versus not. And so that's an interesting question that begs to be addressed, whether these different subtypes are plastic, whether they're interconvertible, et cetera, et cetera, et cetera. And so I will just give you one example that we found early in my independent scientific career, which pertains to the loss of beta cell maturity and impaired identity in diabetes, which is these beta cells that express neuropeptide Y. So what we discovered using pancreatic tissue from the NPOD repository was that both subjects who have type 2 diabetes and type 1 diabetes, their beta cells, the residual beta cells, seem to have a subpopulation that is expressing neuropeptide Y compared to the controls, which do not seem to have this subpopulation. And if you look back in developmental time, neuropeptide Y is expressed in the neonatal beta cells, but is repressed in the adult beta cells. So again, this is a question of beta cell dedifferentiation, beta cell going back in developmental time. And this we published a while ago in JCI inside my really talented undergraduate student, Pope Rodnoy, who's now a physician. He contributed mostly to this important work. And what Pope also found when he used this neuropeptide Y GFP model, that this sort of change of beta cell heterogeneity not only happens in case of diabetes, but also in case of scenarios where beta cells are exposed to stress and they're trying to adapt. So here's a snapshot of neonatal pancreas, where the islets seem to have this population that's enriched for MPY, which is gone from the adults, but it reemerges when you expose beta cells to stress, for example, by way of high-fat diet. And so one thing that's been evident in the field is that all diseases at many levels recapitulate an ontogeny, which is like the developmental context, right? And my hypothesis is that they do so for the purpose of cellular adaptation. Now, if this doesn't pan out, and if the stress continues, then adaptation can at some point get into the zone of maladaptation, and that's when beta cell failure happens. And so I think the context of heterogeneity matters. We can have heterogeneity, say, for example, at a specific developmental state or a specific physiological stress context. And I think it's important to understand that. The other thing is, and this is just a visualization of the neonatal islet that expresses neuropeptide Y. There's something to be said about the developmental regulation of these features and the location of these cells, where they are located, who are they talking to, are they responding to a certain developmental or environmental cue? I think those are the questions that are very important. And this is where my laboratory has taken a lot of interest in. So the theme that we are focusing on is how these cells are developmentally programmed. I mean, what shapes a beta cell phenotype? What shapes a functionally mature, fully functional beta cell phenotype? And the story begins, when you have the formation of the pancreas. So from pancreatic progenitors, you get the endocrine progenitors, which then make these immature islets that are non-glucose responsive. At some point, they change their architecture, they become functionally mature, and they become glucose responsive. Then the body is exposed to a variety of stressors throughout the lifetime. I mean, beta cell is constantly exposed to changing insulin demands, right? And so there's a lot of stress. And so beta cell can fail. So the question is, how is this information for making a mature islet programmed, and when? So we are investing a lot of time and effort in understanding the mechanisms whereby this functional phenotype is encoded. And our MO is to look at the epigenetic patterning of the functional beta cell phenotype using both mouse genetics as well as human samples. And so the reason why we are so invested in the idea of epigenetics is because it's basically the cell's toolbox of reappropriating the same genome in different cell types to different endpoints. And in doing so, it allows a cell to tailor its behavior to the changing environment by interpreting the genome in a very context-specific manner. There's different kinds of epigenetic modifications. There's modifications of the proteins that bind to the DNA. There's direct modifications of the DNA. And we're really interested in understanding how this epigenetic code programs the behavior and the differentiation of a cell during development. And so the one epigenetic modification that I think has been very well studied, the most well studied, and I think one of the earliest discovered, is the methylation of the cytosine in the DNA. And it's regulated by enzymes called the DNA methyltransferases. And so today I will be talking to you a little bit about this work, which was initiated in the lab by Kimi Wang and Nazia Perrine, and is now being continued by a new student in the lab, Gio Hernandez. And so what is DNA methylation? So you can have a de novo mechanism whereby a stretch of DNA that's never seen methylation before gains a new piece of methylation information. And the primary enzyme in islets for that is the DNMT3A. But then once this methylated DNA undergoes the process of cell division, you have a situation. You have a hemimethylated stretch that does not have the information from the parent cell. And so you have another beautiful mechanism in nature that recognizes and replenishes this epigenetic information. That's the process of maintenance methylation. That's regulated by DNMT1. So there's the formation of new information which is instructive. It provides new signals. And then there's the maintenance that safeguards these patterns that are generated through the process of cell division. The question is, how does DNA methylation patterning regulate beta cell homeostasis? My friend Santa Georgia and I, during our training, invested a lot of effort in this direction. So Santa Georgia published this really beautiful paper some time ago where she demonstrated that DNMT1 dependent maintenance methylation in the pancreatic progenitors is really important for the maintenance of the progenitor pool and their subsequent differentiation. Our joint work together demonstrated that this type of maintenance methylation of existing patterns is also important for sealing the alpha versus beta cell fate choices. And if you do not have this maintenance methylation mechanisms, beta cells dysregulate this lock of fate. They start expressing factors that are normally expressed in alpha cells, and they switch their phenotype. And then with Laurie Sussel's group, some of my work with Anil showed that even de novo methylation, new methylation information is important here. And subsequently, we also found that new methylation patterns also are very important in guiding beta cell maturation from a glucose non-responsive to a glucose responsive state. And so the question is, we know all of this about safeguarding the choices between two distinct endocrine cell types, alpha versus beta. We also know about new methylation patterns that direct the switch in beta cell state from an immature to mature state. What is the contribution of these type of epigenetic patterns in guiding beta cell heterogeneity? And so thus began our quest. We focused on a particular cell type that's observed in mice, which is the beta cells that express tyrosine hydroxylase. This is a collaboration with Dr. Santa Georgia, Dr. Benshi, and Dr. Supriyo Bhattacharya. And in this project, we were really interested in these tyrosine hydroxylase positive beta cells. Now, tyrosine hydroxylase is something that's expressed in neurons. Okay, it's a very sympathetic neural-like feature. What it does is it's a key step in the biosynthesis of catecholamines, epinephrine, norepinephrine, which are known to repress insulin secretion. But I'm just intrigued by the fact that these characters are present within beta cells. And it's a known thing, beta cells do appropriate a lot of developmental paradigms from the neurons. And so to me, this was interesting. How is this beta cell heterogeneity created? This cell type, however, is not just prevalent in mice. We also see some of it in humans. And thanks to the available data sets, for example, the one from Powers and Bushova lab and also from the housing lab, we were able to see, yes, you can find adult beta cells that have some of the transcript, but it does not seem to recapitulate in terms of the protein levels. We were not able to find tyrosine hydroxylase positive beta cells in the adult pancreas, even though you have TH positive neurons that prevail in the islets. But if you look at the neonatal pancreas, you do see this kind of TH positive beta cell subpopulation. So compared to the adults, there's some TH positive beta cells in the neonates. So there is some developmental restriction there. And then again, if you look through the available data sets from the Ottenkowski lab and Jan Lee's lab, what we found was that then again, this developmental restriction seems to occur even in the stem cell derived beta cells. So for example, this data set has the early stem cell derived beta cells, the late stem cell derived beta cells, which are somewhat more mature, and the adult human islet derived beta cells. So if you compare them, TH expression is more prevalent in the more immature states. And then Jan Lee's group did this cool experiment where they had compared the islet derived mature beta cells, the beta-like stem derived products that were less mature, and the ones that had seen the ROCK inhibitor treatment, which the hypothesis behind that being that this would improve the maturation. And so you can see that the ROCK inhibitor treatment does seem to reduce the level of tyrosine hydroxylase, again, suggesting that in humans, this is a more developmentally restricted cell type. People always ask me this question: are these really beta cells or some kind of like neural imposters that are sitting within the islets? So yes, they are beta cells. They express all characteristics of beta cells, for example, the transporters, the different transcription factors. So the question is, how are they then different? And they're functionally mature because they have urocortin 3 in them. So really they are standard issue beta cells. So the question is, how are they different? So the first difference that we found was that they seem to have a lot more connections with these sympathetic neurons. So if you see this co-stain with TUJ1, they seem to be more tightly linked to the sympathetic neurons. And this is quantified here. You can see that the TH positive beta cells seem to have more 1 to 2 fiber contact compared to the TH negative cells. Similarly, they also seem to be in close proximity to the blood vessels. So here's a stain for the PCAM1 marker. You recognize the endothelial cells, and you see similar behavior. So indeed, they might have different connectivity to the outside world, if you will. And this can be recognized if you look through the Z space. So this is for the PCAM1. And then if you now want to ask what is so distinct about them, then again, I want to thank people who submit their data sets for public use. This is a data set from Dr. Raghu Mimara's lab. And when we looked at the data sets to see what set tyrosine hydroxylase positive beta cells apart from the tyrosine hydroxylase negative beta cells, what we found was, so here's the TH positive fraction showing the specific gene signatures. What we found was that they seem to be metabolically very active, and they also seem to be very poor at handling ER stress. And Senta's lab helped us figure out that indeed that is the case. They looked at the flame imaging. They use these flame imaging technology to look at the NADH lifetime, and they found that indeed TH positive beta cells are more OXPHOS enriched. And then we also looked at the ARO1B marker, and we found that indeed TH positive beta cells have reduced ARO1B. That again is suggestive of poor ER stress handling, perhaps. And so they are metabolically and molecularly very distinct subpopulation. So what locks this TH expression? So we sorted cells from lineage labeled pancreatic progenitor, endocrine progenitor, and beta cell models. So this was a PDX3 derived YFP. This was NGN3 promoter derived GFP model, and this was a MIP-GFP model. So we sorted pancreatic progenitors, endocrine progenitors, and beta cells. And then we looked at the tyrosine hydroxylase promoter methylation. And what we found was that indeed TH promoter does undergo new methylation information through the process of beta cell differentiation. So as beta cells, as the progenitors transition from the pancreatic progenitor state to the endocrine progenitor state, and then to the beta cell state, we have more and more DNA methylation. So this schema, this line diagram is actually output of a bisulfite sequencing experiment where you are able to distinguish between methylated and unmethylated Cs, which are represented with filled circles and open circles. So you can see that in beta cells, TH promoter is more heavily methylated compared to the progenitor states. So this suggests that the methylation might be happening somewhere between the transition from endocrine progenitors to making beta cells. So how do we test it? So the way we went about it was we were fortunate to have the models that lacked the new methylation enzyme DNMT3A either in the pancreatic progenitors or the endocrine progenitors or in beta cells. So let's take these 3 models, look at their pancreas and see what happens to the tyrosine hydroxylase expression. So what we found was that loss of DNA methylation in pancreatic progenitors and endocrine progenitors led to dysregulation of tyrosine hydroxylase expression. But really nothing happened when you lose DNA methylation in the beta cell specific knockout. So this again suggested the same thing, that the methylation must be happening somewhere during the transition between the endocrine progenitors to the beta cells. So there is a very developmentally specific thing that's happening here, right? And so you can quantify these cell populations, and then you can look at the methylation patterns and indeed recognize that you do lose this new methylation information when you lose the NMT3A in the progenitor states, but not in the beta cell states, again confirming the fact that methylation must be happening between the endocrine progenitor and the beta cell states. So that's for making new patterns. Is it important to maintain them? So that's the idea of maintenance methylations, because beta cells, of course, once they're formed, they do undergo replication in the early stages of life, and they can sometimes undergo replication even in late postnatal stages if exposed to stress, right? So is maintenance important? So for that, we have this really fruitful collaboration with Center, where we had these tissues available from the Georgia lab, where we looked at the DNMT1 knockout in the endocrine progenitor states, which is the failure of sustaining this methylation information. So earlier we were looking at the failure of making new information. Here we are looking at the failure to maintain information. And indeed, failure to maintain this epigenetic information does result in a collapse of TH restriction. You can see the upregulation of tyrosine hydroxylase. Same is the case when you delete DNMT1 and beta cells specifically. So unlike the new methylation information, loss of maintenance methylation in beta cell does alleviate this restriction. And the reason behind that is you don't have the new methylation reactions happening in the beta cells, but you still need to maintain what happened before in the development. And so if you take islets from these beta cell knockouts of the maintenance methyltransferase and compare the tyrosine hydroxylase methylation, indeed you see that the knockouts have reduced methylation. So failure to maintain this methylation regulation is really what translates into loss of this beta cell heterogeneity, if you will. So what's the physiological relevance of this, if you may ask? Now, before I go into that, I just want to highlight one thing. I think, so this is a new unpublished piece of data set, and I just want to show something there. So what we've done now is that we have a single cell sequencing atlas of these DNMT3 knockout pancreata. So now we can go in and look at specific cell clusters and see how their identities are being impacted. So for example, here you can see this neural-like marker is only getting dysregulated in specific clusters compared to the controls. TICSNP, which is a stress marker, again, is only getting dysregulated in this specific cluster of beta cells. So you can now go into these kind of data sets and ask very specific pointed questions about different types of heterogeneity, if you will. Now, coming back to the physiological context, what happens to this type of heterogeneity or the epigenetic control of this type of heterogeneity? So what we've done here is we've taken animals and exposed them to high-fat diet protocol for about 16 weeks, which is the long-term high-fat diet, which pushes them to the brink of beta cell failure. And what we observe is that in the failing islets, you do have loss of this epigenetic restriction of tyrosine hydroxylase due to the loss of this methylation, suggesting that indeed maintaining this methylation in the physiological context is really, really important. And so that does tell us something about how beta cell heterogeneity is encoded and maintained in the lifetime of beta cell. Now, the other important question is, what about other cell types? So I find tyrosine hydroxylase to be very interesting in that in the early embryonic development, tyrosine hydroxylase sits in the alpha cell domain. Okay, and then in the postnatal states, it not only is there in beta cells, but also in a subfraction of delta cells, but it's completely gone from alpha cells. So the question then was, well, if we break TH restriction, epigenetic dependent TH restriction in early development, does it only affect beta cells, or does it affect alpha cells or delta cells as well? Which ones and when? right? So as I mentioned, a subfraction of delta cells also expresses tyrosine hydroxylase. And if you look at our single cell data set from the knockout, you can see that tyrosine hydroxylase is upregulated in the knockout delta cells. And you can again look at the knockouts by a histological method, and you can see indeed TH is upregulated in these knockout delta cells. But we were not able to find any alpha cells that dysregulated tyrosine hydroxylase, even though the embryonic alpha cells seem to express a lot of tyrosine hydroxylase, suggesting that this step is slightly later in development. This restriction of tyrosine hydroxylase is in the beta and delta cell population is regulated distinctly compared to the regulation in alpha cells. And so what are the remaining questions? Of course, this is one method of controlling beta cell heterogeneity. What do other beta cell subtypes, how are the other beta cell subtypes controlled, right? We don't know that. There's a collaboration that we have now with Dr. Steve Millership and Guy Rutter's lab, where they're looking at neonatal regulation by DNA methylation. So that's another cell type that might be regulated by DNA methylation dependent restriction. But not all subtypes may be regulated in the same manner. And the other question is, what are the signals that are responsible? So for instance, is interaction with neurons or blood vessels is something that shapes. So Marcela Bersoa's work has shown very beautifully that when they alter the VEGF signaling, that leads to altered vasculature, and that leads to massive upregulation of these TH positive beta cells. So maybe there's some link between the signals that the islet receives and how the epigenetic control works. Right, so those are important questions that remain to be answered. But the take-home message that I have here for this talk is twofold. One, that things that happen early in development, for example, this DNA dependent patterning, can impact beta cell future. It may not just impact what's happening in the immediate developmental time, but it can have long-lasting impacts, and that's important. The second thing I think is the cool ever type of thing, the similarity between beta cells and neurons, and that really fascinates me, because beta cells have all these neuronal-like features that are very developmentally restricted. So what exactly are they doing there? I think that's something I really feel excited about and want to continue pursuing. And so with that, I would like to thank Art, who has always been a great champion and has been inspiring and continues to inspire. My team, Sneha, is relatively new in the lab, and she's already charting new territories in the lab. She's a CERN postdoctoral fellow and doing really excellent work on DNA damage response. Pope, very proud of him. Nazi as well. Gio is again a new colleague. Gibby is doing well at Pfizer. Really grateful for my mentors and collaborators, Dr. Kandil, Dr. Thurman, Dr. Raman Natarajan. Rupi and Ben's labs have been great collaborators, as have Senta's and Mesh Yadav's labs from USC. Really appreciate the collaboration with Steve. And so also very grateful for the financial support from all of these agencies. And with that, I would like to thank the audience and take any questions. Thank you.
Thank you so much, Sangeeta. That was a beautiful talk. Really walked right through the entire history of the literature, as well as sort of like the hypotheses and the asking people to think further into the future about what this may mean. I want to open it up to people in the audience. If you'd like to ask a question, please unmute yourself.
Let me stop sharing so we can see each other.
Sure.
Lovely to see familiar faces in the crowd. Hi, Senta, Matt.
That's great. And I think I'm seeing someone. Yeah, Matt, would you like to ask a question?
Yes, yeah, good to see you as well, Sangeeta. Wonderful talk. So I was wondering, the data that you showed on the high-fat diet causing upregulation of TH and demethylation of its promoter, how specific is this towards TH relative to other genes that are regulated through maintenance DNA methylation? Do you generally see upregulation of disallowed genes and other genes that are normally repressed through DNA methylation in response to a high-fat diet? Or are there some genes that are more susceptible to being reactivated?
I think it might be a very graded phenomena. Certain things do come up earlier than the others. For instance, so in this paper, we had this paradigm of short-term and long-term high-fat diets, 8 weeks and 16 weeks. And the shorter term did not induce tyrosine hydroxylase while the longer term did. But neuropeptide Y comes up very early on. And I don't know if that's regulated by DNA methylation or not. That's epigenetically regulated for sure, but I'm not sure if it's regulated by DNA methylation. I know Steve Millership has some data in the making on how high-fat diet breaks the imprinting of neuronatin. And so that's another thing of interest. And I think there's a lot of data on how nutrients can change the methylome. I mean, you more than anyone knows that the nutrient homeostasis is very intimately linked with the epigenome, right? And so depending on the nutrient composition and the extent of nutrient exposure, I think these cells may, upon chronic overnutrition, switch their epigenome to a very dedifferentiated state. I think what we want to do is explore the single cell data sets that are available in high-fat fed animals to see what other neuronal-like markers come up and what is common with our DNMT3A knockout data set. So I think that will answer the question that you just asked.
Great, thank you.
Great answer. Okay, I thought I saw someone else. You can raise your hand if you need help unmuting, or just unmute yourself.
I think Khati Girder has a question, a brilliant question, Khati. Would you like to comment on how immune cells react to different heterogeneous populations of beta cells? So,
yeah, great.
Emerging area in the laboratory. We do have some new data on our DNMT knockout. So, where we see that loss of heterogeneity does lead to altered interaction with the immune system. It's preliminary, so I didn't include it, but here's the question we must ask. Like I said, nothing happens in biology without a reason, right? So this restriction of certain subtypes that happens in developmental time is there for a reason. What would happen if those cell types were allowed to linger beyond their supposed time into postnatal life? Would they interact with things differently? Now, I must say that the neonatal pancreas is very different in terms of the kind of niches it has, right? Like there's a lot of macrophages, et cetera, et cetera. The vasculature is very different. So a lot of these immune-like signals might also be growth and developmental signals back then. So if these cells are allowed to persist, they might alter the interaction of the islet with the immune system. And I think that's a very important question to ask. What is the significance of development restriction of these specific subtypes? You know, you don't want to see these any longer. And of course, they can also entrain the nutrient homeostasis in certain times. For example, you know, neuropeptide Y is a hormone, well-known hormone in the feeding fasting circuit, right? So that's one thing. But what do these cell types do in the neonatal pancreas, and why are they not welcome in the mature pancreas? So for example, some of these cells interact preferentially with the blood vessels. Allowing overexpansion of these cells may alter the interaction with blood vessels, and that could alter interaction with the immune system, right? And so that's a really important questions to think about. So I thank you for bringing it up.
Thank you, Khati. That was great. And just to tap on her a little bit, that she's in the lab of Emerald Dennis in Boston College. They're focusing on the microbiome. So they have some really interesting data as well as to how that interaction may be occurring. Any other questions? I guess I would say, you know, what are sort of, you know, what are your next steps for, you know, pushing this forward? I don't know if you want to give away your any secrets, but you know, where are you going?
Like I said, this is an emerging interest in the lab. The other interest is looking at how cells protect themselves from DNA damage and other kinds of damage in the process of their day-to-day combat, right? They're literally combating a lot of stressors every day, changing nutrient environment, different, you know, levels of obesity, et cetera, what have you. So how does the beta cell make sure it's stealth and robust and survival competent? I mean, beta cells are mostly one and done, right? They don't have a lot of scope for expansion in the adult life. And so you really have to be very robust in terms of protecting yourself against any kind of nasties. So how does the beta cell achieve that? What is the shield? What are the shields?
Mm-hmm.
So that's an area we're really interested in.
Yeah, that's a fascinating area. Let me just check to see any more questions. One more thing is how has anyone looked at, you know, post people who have had type 1 for a long period of time and what their TH looks like in context there?
You mean in the serum or in the pancreas?
Pancreas. But I mean, maybe the serum is interesting also.
So we've looked at the type 2 diabetes pancreas, and again, we did not see very much going on, but there's hurdles to that because I think it's also it's the human tissue is not collected exactly the way we collect tissue in you know our models, right? And so the processing time, et cetera, varies, and so there can be discrepancies that, and plus we cannot really accumulate a large n, a very large sample size. So I think just because we did not see something in 2 or 3 samples from a certain disease state does not mean that it's not there. I think that's where the power of single cell data sets that are coming in from the consortia like HFAP will come in. And so we are on the lookout for like appropriate data sets. We are willing and going to generate some of our own in that regard and look through to see. I'm really interested in the reappropriation of neuronal characteristics in beta cells.
Yeah.
So that's something really fascinates me.
Yeah, it is very fascinating. It's still not very well known. So, okay, last call for in interest of time. I know we started a little bit late because we were adding people in. Last call for question, or you can reach out to Sangeeta Dhawan via email. She's located at City of Hope. And I guess my last question is, do you have any postdoc positions available?
We're always looking for amazing scientists to work with. So just send an email.
Great. Send an email. I mean, it's a fascinating field. Really, really interesting. Cannot wait to see what next comes out of the lab at City of Hope. Thank you again for joining us. And congrats on some beautiful work.
Thank you. Thank you, guys. Lovely to see you. Bye.
Bye-bye. Have a great day.